Hey class, so I'm going to give you a quick lecture on the lymphatic system. Alright, lymphatic system. There are three major functions on the lymphatic system. One function is water balance, another function immunity, that's a very important one, and another function is basically returns lipids in the circulation, absorbs lipids, transports lipids, not really absorbing but actually transporting lipids, right? Lipids. Right, but I want to quickly discuss water balance and the immunity to some extent on the lymphatic system. There's a circulatory system. That's your blood. Blood is the highway. So anywhere you enter, a particular nutrient, chemical would be delivered to any parts of the body. Anywhere you scratch yourself, you're bleeding, that nutrient will be delivered there to a particular cell. Lymphatic system is a one-way route or the road. So think of this as the highway, the Bell Parkway, and this one is a service road going one way. So it's basically lymphatic system picks up leftovers and tries to return it back in circulation. So here's the blood vessels, this. Here's your interstitial space. Here's your cell. From a blood vessel, we want to deliver nutrient to the cell. It needs to cross this interstitial space so we can deliver it here. And the waste goes in the opposite direction. But what if a particular nutrient like protein and water kind of gets stuck in interstitial space, in between? It cannot go back in the circulation. So here comes the lymphatic to the rescue. So the lymph vessels, lymph capillaries, they're basically running through the interstitial space and they're able to return water and the lost chemicals back, not back, pull them into the lymphatic system and then eventually return back in the circulatory system. So basically, if you have this lost protein, we will take it into the lymphatic system and we will return it back in the circulation. Same thing happens with water. If there's lots of water, to prevent edema, we can also bring it back into the lymphatic system and return it back into the circulation. So now that you know the big picture, let's look at small details. So here, this represents a capillaries, right? Here, let's say, are arterioles, right? And these are venules. And cells are here, right? So we're bringing that chemical, want to bring in the cell, but it just got lost, it got stuck. And here are the capillaries. So basically capillaries of the lymphatic system, they are all over the place and they pulling that particular nutrient or the water and the waste as well. So that's the one major function that we just discussed. It was water balance, right? So now let's discuss immunity. What if we have an injury? What if the bacteria or the virus, any type of pathogen enters your system? Not necessarily through the damage of your skin, but through the oral cavity, nasal cavity, sexual reproductive organs. So that can also end up here in interstitial space in between cells. So what happens that lymphatic system can pick this up as well, but will not return back in circulation. So now let's look at lymphatic vessels. So here are the lymphatic capillaries. They're able to pull water inside because in interstitial space there is a high pressure. Within the capillaries, it's low pressure. So because of the pressure, it is easier to push water and chemicals, nutrients, waste, bacteria, viruses into the low pressure vessels, going from high pressure interstitial space. So the cells of the endothelium of lymphatic capillaries looks like this. Right? So it's easier for the water and the big proteins and chemicals to enter. And plus, in interstitial space, again, that's the high pressure compared to what's inside. So it's getting inside. It should not get outside. And that way we can return water and all of this crap that sits in between the blood vessels, between the cells with the interstitial space. So once we're able to pull that within the capillaries of the lymphatic system, now we want to return back in the circulation, right? Lost protein, for example, water we needed, but what about those pathogens? So here's lymphatic capillaries, and there's the lymphatic vessels. And while, well, because it's a one-way system, and that's a circulatory system, while we're trying to return it back, so that lymph is that fluid, right? Lymph is that interstitial fluid that will pass through the lymphatic vessels. And we have here our checkpoints. They call them nodes of the lymphatic system. Within those nodes, there's a lot of information here, but within those nodes, we have aggregation of immune cells. Those are T cells and the B cells that can flag the pathogen and mark it for the destruction. So there's a lot of nodes. Like if you look at the capillary system, so let's say these are the vessels, and you have these nodes, right? A lot of nodes in the lower region of the body, in the thorax, in the upper region, in your upper extremities, lower extremities, in your neck, in the sexual reproductive organ, in the head and the face, pretty much throughout the entire body. Because we need to return water, and we need to return big chemicals like proteins that got lost, and the bacteria. So we need to check, we need to double check if this really belongs to our system, and we want to cleanse it. So think of that as like a sewer and a cleansing system. So let's say that chemical gets in here and the water gets in here, and lymph node's job is to make sure that there are no pathogens, that everything belongs to self, meaning that it belongs to our system. If something is being tagged or has a, let's say, non-self antigen, that will be marked for the destruction by the immune cells. There are things that I'm skipping for now. I will go back at the end of this tutorial to the T cells and B cells, self and non-self. So, and it goes through this bunch of nodes while it's traveling, so we can return it back into circulation. So basically, if this is the human body, right? I'm a great graphic designer, so we try to return the lymph in two places. Here's a subclavian vein, here's a subclavian vein. There's the left subclavian vein, this is the right subclavian vein. So we can return lymph in one of those. So everything that I'm shading, right, and there's a head as well, right, here's the neck. So all this would be returned into the right subclavian duct. waste, damaged cells, cancer cells that were picked up by the lymphatic system would be returned through the superior vena cava into the heart. Right? Let's say here's the heart, like this, right? So it would be returned into the superior vena cava. The rest of the body, left side, left side, left side, left side of the abdomen, slightly to the right as well, right limb and the left limb, everything would be returned through the thoracic duct into the left subclavian vein. So the thoracic duct will run here, right? And then we will return into the left subclavian vein. So that would be a thoracic duct. That's what I mean, we will return the lymph back into circulation. Now let's talk about T cells and B cells and the lymph nodes. So let's talk about immunocompetency. I'm sorry if I made a spelling mistake, right? Immunocompetence. So there are T cells and B cells. So both of them are made in the primary lymphatic organs. So the primary lymphatic organs are bone marrow, bone, right? And a thymus. So the T cells and the B cells, immature cells, right? So they made in the bone marrow. Made in the bone marrow. So here's a bone. Again, I'm a great graphic designer. That's how I draw the bone. So the T cells and B cells both made here. The B cells, specifically B cells, they mature here. B cells, they mature in the bone. They become immunocompetent in the bone. The T cells, also born here, so born, right? T cells also born here, but they become mature or immunocompetent within the thymus. So basically, you have a bone here, right? T cells and B cells made in the bone. T cells goes in the thymus. It's a bilobe organ. Thymus. And the B cells mature here. So the thymus, we need it for T cells maturation, and we need the bone for the B maturation, B cell maturation. After they mature, right, both of them, T cells and B cells, will end up in a lymphatic system, right? I'm going to put note here that represents a lymphatic system. And eventually, the mature T cells and B cells will also end up in a circulatory system. T cells and B cells made in bone, T cells mature in the thymus, B cells mature in the bone. Both of them will end up both of them will end up in the lymphatic system, and then eventually they will end up in your circulatory system, basically in blood. So now you see when a lymph is moving through these nodes, checkpoints. Here we have high aggregation and multiplication and proliferation of the T cells and B cells. So that chemical, water, waste, nutrients, let's say protein, I'm using protein because it's important, bacteria, pathogens, I'm going to put pathogens, bacteria and viruses, they all eventually, right, end up here, and they end up in, and they're passing through the lymph nodes, and they would be checked, purified, they would be checked, are you belonging to our system or you do not belong to our system? So the pathogens, especially pathogens, they have antigens, right? We discussed what the antigen is. But the antigen is the molecule that's sitting on the cell membrane that can stimulate the production of immunoglobulins, of antibodies. So this pathogen would be marked for the destruction, then these immune cells will know to destroy it. I'll quickly will explain the pathway and the process, but I'm not going to go very deep with this. Very simple explanation where I will skip a lot of steps. So what happens? The T cells, there's a helper cell, there's a memory cell, there's a cytotoxic cells, but big picture. So the T cells can recognize the pathogen. How can they recognize the pathogen? So let's say we have antigen, it could be bacteria, it could be virus, right, that can stimulate production of antibodies. We have cells, we call them antigen-presenting cells. These are the B cells, these are the dendritic cells that sitting or circulating or sitting right under the skin, for example, or in the entrance into the host, right? So they can grab the pathogen and kind of present it to the T cells. So it's basically on top of this presenting cells, you have something that is called major histocomparability complex. Histology. So it's basically this cells are phagocytic cells. They take this pathogen inside of them, they will break it apart and will take the sequence, the DNA sequence, and they call it epitope, and they will place it here. So the immune cells, like T cells, can take a look at this. Do you belong to my system or not? The one thing that I didn't mention, when the T cells become immunocompetent within the thymus, they've been presented with the cells that belong to our system. So they want to be with 100% assured that the particular presented chemical actually belongs to our system. So it's basically out of the 100 of the T cells that end up in the thymus, look at this like a boot camp, would be presented with the chemical. And they will say, yes, it belongs to my system, self. So they can recognize. If they will fail at least once, they will be destroyed. Only 2% will survive. And this 2% with 99.9, let's say 100, I'm not really sure, percent can ID molecule cells that has the particular flag that says, it's hypothetical flag that says self, I belong to our system. Otherwise, there could be an autoimmune problem, autoimmune disease. So, these T cells, look at this epitope, it's just basically degraded pathogen that has a particular sequence that unrecognizable, that does not belong to our host, and they can say, is it self? If it's not self, then it is a non-self, and would be marked for the destruction, would be tagged. So the T cells will stimulate the B cells. I'm skipping here, there's the plasma cells, but the B cells eventually will stimulate the production of antibodies, immunoglobulins, like antibody, not a receptor, but like antibody, like that. And this antibody were developed through this particular T cell against this particular pathogen. So if there's two different pathogens, so there's going to be two different antibodies. If there's 20, there's going to be 20, and so on. So the T cells could be memory cells, cytotoxic cells, but the memory cells, meaning that it will be sitting within our system for a long period of time, even decades. There's research on that. That the next time when the same pathogen enters our system, you do not have to do all this process from scratch, because you already have in its database this particular thing. So like, for example, if I'm typing something and I have the original, all I have to do is just put in the Xerox, and like within 10 seconds I have 20 copies. Otherwise I have to type it from scratch. Or this is doing it from scratch because you've never seen this before. Now that you've been exposed to a particular pathogen, so to the memory cells, right? So you can quickly start making new antibodies, because the antibodies can disappear from the body. It depends on how fast. But the memory cell sits within our system for a long time. Same thing with the B cells. B memory cells. So this is very quick flow of immunocompetency, lymphatic system, and the water balance.